Hi learners, welcome back to our molecular biology series. Today we are learning DNA fingerprinting, a technique that uses DNA to identify people just like regular fingerprints. It's used in crime investigations, paternity tests, and identifying dead bodies. Let's see how it works. Humans are 99.9% identical, but that small 0.1% difference is actually huge when you consider the scale of our DNA. Since the human genome contains 3 billion base pairs, 0.1% of that number equals 3 million base pairs that, that vary between any two people. These 3 million differences are what make each of us unique. However, comparing all 3 billion letters to find those specific changes would be nearly impossible. DNA fingerprinting gives us a shortcut. Instead of sequencing the entire genome, we focus only on specific highly variable regions like VNTRs and STRs to identify individuals quickly and accurately. The secret of DNA fingerprinting is repetitive DNA, regions where a small sequence repeats over and over, like the sequence GATA. It is repeated hundreds of times. These are called satellite DNA or variable number of tandem repeats or VNTR in short. Here is the key: the number of repeats varies between people. You might have this GATA sequence repeated 50 times, I might have it repeated 70 times, your friend might have it repeated 100 times. So these vary, these regions are highly variable among two individuals. These regions don't code for proteins, so mutations here do not harm us. They just keep accumulating and creating variations. These variations are inherited. We get patterns from both parents, which is perfect for identification. These type of variations are known as DNA polymorphism. It arises from mutations that occur in germ cells. If these changes do not affect a person's ability to have children, they get passed down and spread. In a population, if a specific mutation occurs with a frequency greater than 1%, it is officially classified as a polymorphism. Non coding regions like satellite DNA accumulate these mutations much faster because they aren't under selection pressure, meaning changes here do not usually affect survival. These include VNTRs or mini satellites or STRs known as short tandem repeats or microsatellites. STRs are even smaller than VNTRs. So these variations pile up generation after generation, creating unique patterns that serve as the basis of DNA fingerprinting and genetic mapping, eventually providing the raw material for evolution and speciation. This method was developed by Sir Alec Jeffreys in 1984, and here is how it works. The first step is to extract DNA from any tissue. It could be blood, hair follicle, saliva, semen, or even a single cell. Next, to cut that DNA with restriction enzymes. Then separate the fragments by size using gel electrophoresis. In the gel, smaller pieces of DNA will travel farther due to low molecular weight. So the pattern on the gel will be like smaller pieces at the bottom and larger DNA pieces on the top or at the middle. The next step is to transfer these fragments to a membrane. This is known as southern blotting. Fifth step is to add a radioactive VNTR probe that binds specifically to that repeated region that we are looking for. These VNTR probes are synthetic labeled DNA segments used to detect specific repetitive sequence in a person's genome. After we cut the DNA and separate it by size, we cannot see the specific fragments that we need among the millions of other fragments, right? So the solution is we add VNTR probes, which are complementary to the repeat sequence. Because the probes are radioactive or fluorescent, they can be visualized on the X-ray film or auto radiography, revealing a unique pattern of bands. This is how identification is done. So the next step in is detection of bound probe using autoradiography. It creates a pattern of bands, and each person's pattern is unique. To understand how DNA fingerprinting is applicable in crime scenes, we are drawing a crime scene here. So this is our agarose gel. In this first lane, DNA recovered from the crime scene will be running, and the second lane is for a suspect A, who could be the criminal, or suspect B. Either A or B, they can be the criminals. So the DNA was recovered. This DNA was recovered from a hair strand found in the scene, found where the incident took place. So the people from the forensic department took the hair strand, they extracted DNA from it, and then they used restriction enzymes, and now we have DNA fragments that will be run on the agarose gel electrophoresis by gel electrophoresis. So this DNA was recovered from the crime scene. So here is the pattern. And these samples taken from A and B were taken via buccal swab or cheek swabs, because VNTR patterns are inherited and highly unique to individuals. Matching the pattern is considered strong evidence of their presence at the scene. So these are the DNA that we got from the person 1, and this is DNA from suspect B. Small fragments will move quickly through the gel spores, ending up at the bottom. They are like small cars in a traffic. They will go through the gel easily and end up at the bottom. On the other hand, large fragments like this, and this, they are like big trucks. They get stuck and move slowly, staying near at the top. So if you look at the patterns, crime scene DNA and suspect B's DNA, they have identical band patterns. If you look here, they are matching, their bands are matching. So that tells that suspect B was at the crime scene. So this is how they solve these problems. And now, how does DNA fingerprinting solves the paternity dispute? Let's look at that. So Again, here is the gel. A child's DNA fragment a DNA fingerprint is compared to both the parents, mother and father. In this case, suppose this man who is the alleged father, it says that he's not the father of the child. So uh, the DNA samples from all three of them will be taken, and then they will run using the same concept, using the same methods, DNA extraction, and then restriction, digestion, and then gel electrophoresis. They will take the child's DNA, so the child's fingerprint is compared to both parents. Because as the child inherit exactly 50% of their VNTR from mother and 50% from father. Since every specific band in the child's DNA profile must match a corresponding band in either the mother or the father, any band which is not from the mother must match the biological father. This makes DNA fingerprinting a foolproof tool for settling parental disputes. So, in our case, these are the bands of the child, and now these this is the band from the mother. So, one more important thing, these DNA fragments, when we place them in gel, agarose gel, DNA is negatively charged. So, it will move towards the positive end. That means towards the bottom of the gel. So, these two band patterns are from mother, and these two bands are from father. So if you look closely, this man is certainly the father, because these bands are matching. Let me use another color. So these two bands are matching. So the child got these bands definitely from the father. So this is how gel electrophoresis is used in paternity dispute. So that means if we see the inheritance pattern, by looking to the inheritance pattern, we can prove the paternity. This slide is a recap of what we just learned in our drawing session. Here is a crime scene, and blood is collected, and police have two suspects, individual A and individual B. So, how do we find out who was really there? We use their DNA, because these two suspects have different numbers of VNTR repeats. Now, if you look at the gel carefully, individual A's bands are here, that is one pattern, and individual B's bands are also here, which is completely different pattern. Now, look at the crime scene sample. Which one does it match? individual be, right? Every single band lines up perfectly. So it says that individual B was at the crime scene. This is the power of DNA fingerprinting. It tells you the truth with near certainty. Now, let's explore the diverse range of applications of DNA fingerprinting. It has many uses in forensics. Apart from forensic identifying suspects in crime and paternity testing, this technique has many more applications. It identifies victims in mass disasters and natural calamities. When bodies are damaged in case of mass disasters such as earthquakes, wildfires, hurricanes, or man made disasters such as oil or gas leak, plane crashes, etc. When bodies are often physically unrecognizable, DNA is extracted from relatively stable tissues such as bones and teeth. Since DNA remains intact even when other tissues decay, scientists compare the VNTR and STR patterns from the remains of to those of living relatives. They extract the DNA from bone and teeth and then do the VNTR analysis and compare it to the living relatives of the dead people. And after that, because a child's profile is 50-50 combination of their parents' bands, forensic experts can confirm an identity with near absolute certainty by matching these unique genetic barcodes. DNA fingerprinting is also used to study population genetics and diversity. By comparing the frequency of DNA polymorphisms between groups, they can track how populations move from one place to other, adapt, and evolve. It shows us the genetic distance between two different groups of the same species. It also helps in wildlife conservation by tracking endangered species. For endangered animals, researchers use DNA fingerprinting to prevent inbreeding. Inbreeding is when two individuals who are closely related, like family, they have offsprings together. And it is not good for the gene pool because the chances of inheriting the damaged genes is increased in inbreeding. So, by identifying which animals are too closely related, they can choose the best pairs for inbreeding to keep the gene pool healthy and diverse. It is even used in immigration cases to prove family relationships, all from analyzing those repetitive DNA patterns. When people move to a new place, new country to join their family, governments often require scientific proofs of their biological connection. Since we inherit exactly 50% of our VNTR from mother and other 50% from our father, a DNA fingerprint acts as a genetic passport. By comparing the repetitive DNA patterns of the applicant with the relative already in the country, scientists can prove a parent child or sibling relationship with nearly 100% certainty. If the band in the child's DNA don't match the claimed parents, the family relationship is disproven, making this the ultimate tool for solving legal immigration disputes. Now, here is the quick summary of what we have learned so far. This method of DNA fingerprinting was developed by Alec Jeffries. It identifies individuals by analyzing unique, highly variable DNA sequences called VNTRs. The process involves extraction, digestion, separation, and hybridization. It creates a distinct genetic barcode used for identification and forensics. And for NEET, you must remember Humans are 99.9% identical, but the 0.1% difference in our repetitive DNA shows high polymorphism. Alec Jeffreys developed DNA fingerprinting by targeting VNTRs, where the number of repeats varies uniquely between people. The traditional method uses southern blotting with radioactive VNTR probes and autoradiography to reveal a specific banding pattern. Today, PCR has made it so sensitive that even a single cell is enough to solve crime or settle paternity disputes. Remember, only identical twins share the same DNA fingerprints. That concludes our discussion on DNA fingerprinting. We have now completed the chapter Molecular Basis of Inheritance, with the remaining sections to be integrated into our forthcoming cell biology series. Moving forward, we will begin Unit 9, which has Chapter 9 and 10, focusing on the principles, processes, and applications of biotechnology. This unit serves as a logical progression from our recent study of Human Genome Project and DNA fingerprinting. So I look forward to seeing you in the next video. Study hard and take care. Thank you.